Okay, welcome back. Our next speaker is Randy McIntosh from the University of Toronto, the Rotman Research Institute there. And he's going to talk to us about moving from mental states to mental processes via connectome-based brain simulation.
Yeah, quite the title. Well, thank you, and thank you again for the invitation. I'm actually quite inspired by both the topic as well as the speakers that have come so far, as well that will come in the future as well. My presentation is not going to talk so much about ways of making a new ontology, but rather think more about the metrics that one might consider in making that mapping, particularly having to do on the brain side of things. In the talks you've heard thus far, there's been a lot of focus on, I'm not going to use locations in sort of the local adjacent perspective, but sort of where things are happening, as opposed to when, for example. And the mapping between the cognitive processes that some of us are interested in studying and the physiological processes have to take into consideration that in the brain, there's a space and a time structure that's quite important for the function of the system. And from my perspective, that's actually a very important consideration to try and figure out the mapping between the phenomenology of cognition and the biophysical dynamics that we're measuring with fMRI, EEG, as well as other techniques that we use in neuroscience. So the game plan is to first of all just get a broad overview of what I mean by spatial temporal scales in the brain and talk about some of the important features that are inherent in talking about a space-time structure. That being, for example, there's noise in nonlinear systems that's quite important for enabling certain level functionality. And then ways of measuring the capacity of those systems to be able to integrate information, for example, using entropy measures. We'll then dive into some modeling of those dynamics using space-time structures and look at network switching, not cognitive switching, but network switching. And then move into some empirical applications that take into consideration that where and when things happen in the brain is quite important understanding how it's working. So scales in the brain, you can look at any neuroanatomy textbook or neuroscience textbook and find out that there are a number of levels at which the brain functions. And I would say there's no one particular level that's more important than another. It's actually more important to understand what you're trying to map onto that function. So for example, you can go from microscale circuits that are the local populations we've heard about already today. You can look at the mesoscopic scale, which is getting closer to what we're measuring, for example, with fMRI and putatively EEG or MEG. And then the very macro scale, which we're actually measuring with things like fMRI. Each one of these scales can have multiple time scales over which the constituents in that scale are actually interacting. So local populations, for example, in the microscale will interact at a much faster time scale depending on how the proximity of the entities. As they're further away from one another, the time scales change, and therefore the spatial temporal signature changes. You can see that if you scale it up to an entire brain. So for example, on this side you have the entire connectome, if you will, in a human brain. You have local populations that are interacting at a very fast time scale relative to the other populations that are more distal. So here, effectively, the time scale is essentially instantaneous for the purposes of which you can measure in a brain. Whereas here, you've got time delays that are imposed by the fact that these areas are far apart. So obviously, it takes longer for a signal to propagate from parietal to frontal cortex than it does for signals to propagate within frontal cortex. And that time delay is going to be important for how signals are propagated and how information is therefore integrated in the system. So systems that have this complex structure, and complex here I'm meaning in terms of complexity, will also generate noisier signals because of this space-time scale. And that space-time scale, that separation of time scales across spatial scales, results in a noisier signal that represents the capacity of that system to both process information currently, but also encode new information in the future as well. This is a very simple cartoon showing you, for example, a lattice arrangement here where putatively the signals, because it's a redundantly connected system, would be fairly deterministic. You ping a neuron or a unit, for example, in that system, you get a very simple sinusoid, and no matter what you ping, you get the same response. It's a very robust system because you can knock out a node and it'll do the same thing again, so it's great for that, but it can only do a sinusoid, which is kind of boring after a while. Whereas a more complex system with a more complex geometry allows for a more interesting dynamics, if you will, interesting information representation, and you'll capture that in the signal of the system as well. You can show this in a very simple toy example again. And this is capturing something I've been calling a dynamic repertoire of a system. What it means to have noise in a neural context, neural system, pardon me. On the left side, you've got a very simple 3-node network, 3 oscillators that are coupled that has no noise in the system. It's a single attractor, so if you ping the system, you have an initial oscillation, and then the system relaxes to its fixed point, which is in the middle of that, what's called the manifold. So this is a phase space projection, and it's just showing you the trajectory of those 3 units. Functionally, as they're going from here, which is the initial pinging, they oscillate, and then they collapse into a fixed point. So beautiful trajectory, like figure skating on ice, kind of bumpy ice. Figure skating nevertheless, but doesn't do much other than that. You add noise to the system, you do the same pinging, you'll see the same essentially progression from the initial state through to the fixed point, but it's much more elaborate. It's exploring a broader landscape. It's looking at more potential functional configurations. So the noise allows the system to explore what it can do in addition to what it's actually doing. So noise reflects both the actual and potential functional configurations of a system. And in this particular case, we'll talk about it as the brain. We published a review paper a few years back that captured this idea from the perspective of intrinsic activity in the brain, or resting state, as we've heard about already today, and trying to model what it means to have a fairly complicated pattern of intrinsic activity. Kristen showed some nice diagrams from this morning about the resting state networks. There's even more richness if you look at finer timescales as well. The basic take-home message from the paper was that the formation and dissolution of these resting state patterns, or intrinsic networks, if you will, reflects the exploration of possible functional network configurations around a stable anatomical skeleton. So we assume that in the nervous system that there's a deterministic structure that's imposed by the anatomy. It's relative, of course, because the structure does change, for example, with maturation and putatively learning, but in terms of the immediate temporal scale you're looking at, that's more deterministic than the local synapses, for example, which will change much more rapidly than the large bundles that are going to be connecting areas of the brain. So by and large, the anatomy is fairly deterministic relative to the functional dynamics you measure with functional connectivity, for instance. So to test that, you need to find a way to compare, for example, messing with the anatomy and so on and so forth, and seeing if I start making assumptions about the deterministic structure, about the dynamics of the nodes, what do I get? If I change the noise profile, if I change conduction velocity, if I change myelination, so on and so forth. Let's do that. Can we do it empirically? No way in hell. That'll take you a lifetime to do that. Let's do it as a model, shall we? Okay, let's do that. Computational modeling can do that for you. But you need a way to try and impose a spatial temporal structure. And one way you can do that is to measure it directly in a brain. We have beautiful techniques now to measure the geometry of your brain. You can measure the location of areas, you can measure how far apart they are, and in theory, you can measure how they're connected using diffusion tensor imaging, or at least infer how they're connected. So you can use that as a starting point for your deterministic structure. And that's at the core of this platform that we developed called the virtual brain. I'm not going to talk about it too much. You can download it if you want onto your laptops. Not an iPhone, though. Don't put it on an iPhone. But the notion here is to use the combination of empirical data plus the computational theories underlying brain dynamics to fuse them and look at the merger of function and structure that we measure in the brain. The workflow is something like this. You derive some empirical data. This would be the geometry, the location of areas, for example, in the relative distance. You can measure their connections using, for example, diffusion tensor, as I mentioned before. Measure both the distances of the connections as well as some estimation of the capacity or the connection strength, if you will, in big, big quotes. That forms the deterministic skeleton for your system. And then at each node, you add dynamics. This is usually done using what's called mean field approximation, which is basically simulating the operation of large populations of neurons. That can be done either as excitatory and inhibitory pools of neurons or as more phenomenological models, like, for example, oscillators. You can add noise to them and so on and so forth. You can put them in the model directly and just let it idle and generate intrinsic activity, or you can ping it, for example, by stimulating particular nodes, and then generate the same kind of empirical data that you measure in the scanner, whether it be bold activity. You can generate EEG, MRI data, as well as local field potentials to do the mapping back onto the empirical data to tweak the model to have it produce the empirical dynamics that you actually measure in humans or other species. This is an equation because you have to see an equation after having coffee. That shows you a configuration of the model in another framework. This is sort of the general formula for virtual brain. Just saying the population activity at a particular time point is a function of the previous time point, or the change, I should say, in the activity of that population. The influence of the adjacent nodes, so it's the short time scale influences. The influence of the long connections. These ones have the time delays embedded in the connections. There's also an input term here for stimulation, as well as the local noise in the system. So again, local dynamics of the population, its adjacent influences, in a mean field approximation here, and then we have the long range connections that are estimated through, for example, like DTI. And that gives you the deterministic structure I was mentioning to be able to impose the dynamics and look at how these dynamics then manifest themselves in the model. This is whiskey. Who knows the Van Halen? Do you remember that there was so David Lee Roth, who was the lead singer, used to have a bottle of Jack Daniels on the stage, and he would swig from this. We found out many years later that was actually not whiskey, that was iced tea. Pardon me. So, sidebar. So we have now a model that has a deterministic structure and it has time delays in there because of the distances of connections. What that does is sets up a space-time structure of the connectivity matrix. This diagram, the slice to the far left, is the connection matrix between 38 regions of interest based on, this is actually from the COCOMAC database, which is a database of macaque connections in cortex. So where there's a white square that indicates that there's a connection between those two nodes. And that's assuming there's no time delays. If you extend that matrix and add time delays and saying what is possible at a certain time, if you ping a region, what's possibly going to be interacting at a given time point. So for example, the first slice is at 2.7 milliseconds. If I look at signal propagation at 2.7 milliseconds, it's going to be basically local connections. They're going to be interacting, potentially interacting. As I extend that out across from 10 milliseconds to 26 milliseconds, you see that the space-time structure, what could potentially be interacting, changes. This, to me, is one of the most important parts about how the brain is set up. That space-time structure actually scopes what can happen in the system. Not what does happen, but what can happen. So potentiality is quite an important feature of understanding how to map the brain back to behavior. Cool, eh? Okay, so let's try and put this into our virtual brain model and see what happens. So the virtual brain is generating the activity that you would measure if you stuck an electrode into the system. So it's measuring very fast dynamics. Multi-scale representations can be then mapped from the local field potentials to the EEG data to the fMRI data. So that's a different time scale, but also spatial scales as well, local versus global dynamics as well. The way we do this is by putting them all together and then changing the parameters until it produces some interesting stuff. Interesting stuff here is when it stops being quiescent, basically flatlining, and starts producing dynamics that look like what you'd see in an EEG signal or LFP measurement, for example. So here we have what's called the parameter space mapping. This is actually called the stability analysis to look at where does the model become unstable in terms of its dynamics. And what's plotted across the two axes are the velocity of the connections and the coupling strength of the connections in a fairly small model. And as we scale the connection strength, keeping, in this particular case, we keep the noise basically constant, you see that in the upper part of the diagram, you start getting some peaks and valleys suggesting that there's instability there. There's cool stuff happening there. Instability in a particular nonlinear system is good. You want to see that. And what we find is when you get to that point of the instability, what you find, and this is actually the bottom line of that particular trace, is that you have signals that look like what you'd measure from if you put an electrode into the model. So we find an area in the parameter space where the model dynamics look like what you might expect to measure if this was a real head. If we do that in a larger scale model now, one that has both right and left hemispheres, it has a bunch of regions that are geometrically distributed, that have the connections at the right length, and then start pushing it through this stability analysis, we find that the transition from very uninteresting oscillations to more noisy oscillations happens first with the regions that are part of the default mode. And as you move forward in time, these areas pull the entire system forward to where the entire system then becomes entrained and generates these interesting dynamics. And it'll eventually go back to being more oscillatory and separate. But the areas, by virtue of their anatomy, that are the ones that pull the system through this instability seem to be the ones that are associated with the default mode of the network. That's kind of cool. Helps explain a little bit about why this default mode areas show up so much. They may actually be important for allowing the system to change its configurations. Let's look a bit more at the properties of the large-scale human brain, modeled large-scale human brain, when it's in this unstable regime, when it's right at the edge of what's called criticality, edge of what's called the bifurcation as well in terms of dynamical systems theory. So what I'm showing you here is a cartoon that gives you an idea of how we pull this stuff together. Again, you've seen this or heard about it in the last couple slides. You get the tractography, for example, from an individual. You can do this on a single person, by the way. You get their parcellation template if you want to do regions of interest. You can also do voxel-wise modeling as well. Put them together into the structural model there, and then add the simulated brain activity, local field potentials, and then using a forward model, generate the corresponding bowl signal that would come from those particular areas, and then do the mapping between the simulated functional connections, the correlations of activity of the nodes across time, with the corresponding empirical data. And we can scale certain parameters to try and get our fit to be optimal for a particular model. In this particular case, what we're going to scale is we're going to leave the noise at the nodes the same. We're going to leave the distance of them obviously the same. We're just going to change the global coupling of the regions across the system. So again, we're modeling intrinsic activity. There's nothing else going on. The model's just sitting there, lying in the scanner, thinking about supper, for instance. This complicated diagram I'll walk you through now just to give you an idea of what this shows. This top graph is showing you the change in the correlation between different model realizations between the simulated and the empirical data as we scale coupling, global coupling of the model. So you'll see out here where the coupling is weak, the correlations are low. As we increase the coupling, the correlation goes up and up and up and up to about 0.5. And then at this point here, it collapses and goes down. If you look at the attractor space, the manifold, I showed you earlier a manifold, for example, of the system as it traverses across this space, you find initially that there's basically one attractor, a very uninteresting spontaneous state. represents basically the structural anatomy and the functional connections on top of the structure. As you start scaling the connectivity, you have additional attractors appearing, additional networks forming, separate from the main network here. When you get to this point here, this is a bifurcation, so you have a change in the configuration of the system completely. The spontaneous state goes away, it collapses, and you have additional attractors forming. So in this part of the diagram, in this part of the space, this is called the multi-stable attractor space. Here you have a variety of potential configurations available to the system. There's an interesting functional implication of being in that particular domain, in that area of the system. If you look at signal propagation in this model, as you go across that multi-stable attractor space, if you ping the system here, this is looking at visual cortex, for example, and look at the propagation of the signal from V1 to other cortical regions, you find that the propagation speed is rather slow compared to as you get closer to this bifurcation. As you get closer to the edge of the bifurcation, the propagation of the signal increases through the system. All I've done is change the coupling strength globally. That's it. Okay? So by being at the edge of criticality, it seems at that point, the mapping of the anatomy, the mapping of the physiology, the mapping of the noise allows the system to process information optimally when it's right at the edge of that bifurcation. And that edge of the bifurcation is what we use to fit the model to existing intrinsic networks we measured empirically. So that's where your brain is sitting, right on the edge of that bifurcation. And that's important because it allows the system to process information really, really well. Now, I didn't mention things about time. What I've talked about thus far is looking at the potential configurations, without talking much about how the configurations might change across time. Functional connections are, by most measures of the correspondence of signals across time. So usually in fMRI, you take a long time series, say 10 minutes, 20 minutes, and look at the correlation, zero lag cross correlation, for example, across that 20-minute interval. But most of you probably know now, or at least some of you who are doing neuroimaging realize that if you look at smaller time steps, you find that the functional connections in fact change. What's up with that? Well, several years ago, some colleagues who did sort of the neonatal version of the virtual brain did a model, and they found that, yeah, if you do a structural model and look at the functional connections across a fairly long time point, this is actually 240 seconds, you find that the structural connections on the left and the functional connections on the right look quite similar. So function and structure sort of reproduce one another at a long time stamp. But if you look at shorter time scales along the bottom row, you find that the broader functional connections change. So the networks change who they're talking to as a function of time. And that was done in the model, and since then there's been a number of studies that have shown the same thing happening empirically as well. So, and now there's a whole industry that's gone from functional connections to functional connectivity dynamics. So if I take other empirical data and look at the time windows across the top, measuring functional connectivity across a 140-second time window and sliding that forward, you find, and it's hard to see on the slide, unfortunately, you find the functional connections change. And you can actually then look at, do these patterns of functional connectivity reproduce themselves? Do they repeat themselves? This is a very, very broad now characterization of what's happening in the system. If the functional connections don't change, you would expect, for example, the bottom thing to be basically entirely red. Because that would tell you that the functional connections are stable across the entire time. We know that's not true. So what we're looking for now is, are there patterns that repeat themselves? And you find that that's actually happening along in this particular data set. For example, here, as well as here. So the bulbs around there tell you that there are functional connection patterns that repeat themselves. There are states, there are processes that are recapitulating themselves as you go forward in time with this system. It's exploring a configuration, exploring a manifold. You can plot those changes in the functional connections globally in phase space down here and characterize the networks that are actually clustering in terms of states. States here, we're using it fairly loosely, and just indicate a region within this phase space where there are clusterings of functional configurations. What you find is that there are networks labeled alpha here, here, and here that will cluster at a certain portion of time. There'll be a rapid transition, and then a new set of networks will be interacting in state beta. You'll see that indicated in blue. It goes back and forth between these different configurations, exploring essentially a manifold. The manifold here is characterized in a multidimensional space, but suggesting that it's quite important, putatively, for the brain to be able to do that, to explore this broad manifold. Exploring this broad manifold then allows it to consider potential functional configurations as well as instantiate functional configurations, testing predictions, for example, and seeing which prediction is most likely the one I want to try and land on as it traverses the particular manifold, if you will, particular potential configurations to address an external perturbation, for instance. This exploration, this journey across the manifold, generates signals, generates information. So now, can we measure that empirically and map that back to the theory that says that this signal we're measuring should carry information, it should change in some systematic way under certain conditions. So that's the second part of my talk. Can we relate the dynamics we measure to information? So the model suggests that the noise, as well as the anatomy, is an important feature to produce the empirical observations. So we did some mappings loosely back and forth between empirical data and observations. Noise enables exploration of the dynamic repertoire, potential configurations of the system. Noise and information can be related mathematically. So entropy, for example, is a measure of disorder, also a measure of order, also can be related back to noise. So well-behaved noise has high entropy, and I can show you how we're going to measure that in a sense. And we're going to do that with empirical data. So again, getting back to the notion that a system that has more complex structure will generate a noisier signal because it has more information available, both current as well as in the future. Let's try and apply that. This is the first application. This is a study that was published an eternity ago, 2008, looking at the changes in EEG signals in kids in response to faces. This is fairly typical in terms of what you see in the amplitude measures. One thing you notice immediately is that the younger kids have a very high amplitude signal. This is independent of skull thickness, by the way. Very deterministic, it looks like. As you get to adults, you have a more lower amplitude but more multi-componential response in the adults. The question becomes not so much the amplitude, because that's, it's cool but not that cool. We want to see more about the information in that signal. What is the noise in those signals? Can we actually use that to relate it back to what we think is happening in the brain, which is it's getting better in the sense of having a broader functional repertoire. So we're going to use a technique called multiscale entropy, which is basically just there to measure the entropy or predictability or information as a function of different time scales. So what I do with multiscale entropy is I measure entropy at the original time series, and then average it every adjacent point. Measure it again, average 3 points, measure it again, 4, 5, etc. And plot the curve as a function of the time scale that I've downsampled it. So here we have a very non-interesting example of a very deterministic waveform and one that has more information in there. If I do the MSE measures on this waveform, you see that the entropy across scales is fairly low. Here, entropy is high across all scales that I'm measuring here. The scale dependency is quite important in physiological signals that have a combination of deterministic and purely stochastic noise. So for example, if you look at applying the same technique to white noise or 1 over f, white noise putatively having complete randomness in it, whereas 1 over f does have some structure. If I do the same measure with white noise, which is in the triangles, the entropy drops off as I start decimating the signal. As I start averaging points, there's no information in the signal anymore because it's basically random. With 1 over f, there is information across scales. So if I downsample that system, you'll see that the curve is flat, suggesting there's information across multiple scales in the system, which is why being able to do this multi-scale part is important. The time scale is important in physiological signals. So I apply that to these particular data sets. This is an example from, I'm just going to skip that actually. This is an example of the curve. This is from a posterior channel. It's basically the same curve no matter where you look on the head. You see that the entropy for the adults, which is in the cyan on the very top, is quite high across most scales, from fine to coarse scales. For those of you who are into frequencies, you can think about this as high frequency to low frequency information. And then the kids sort of fall ordinally between there, where the youngest kids are on the very bottom, and then you go to the 15-year-olds, and they're in the sort of magenta color. So basically, the information that the brain's extracting from these spaces changes as a function of age. Also, we found that when you correlate these measures of variability back to behavior, that higher noise, higher entropy actually correlates with better behavior across this sample. The top row is the correlation with the stability of reaction time. This is a one-back memory task. You find that the MSE area under the curve, which is the first panel there, as well as a measure of dimensionality using principal components analysis, the higher entropy, the higher the noise is, the more stable your reaction time is in this task. The higher the entropy, the higher the noise, the more accurate you are in the memory task as well. So this noise, this information processing capacity is actually important for good function as well. If you regress out age, the effect goes down a bit, but it's still there. Replicated this in a MEG data set using a slightly wider age range. This is a source model data showing that the strongest difference across the group is actually a change in entropy in the posterior medial cortices in and around precuneus, which is one of the putative hubs in the brain. And also that correlates with behavior as well. This is an awe picture. Come on. Aw. Okay, we're done. You can do the same thing in children if you're really resourceful. Sarah Lippe, who's in Montreal, did this work for her dissertation. 1-month-old infants all the way to 5 years. And she did finish, actually, too. So, but she measured the response of kids sitting on mom's lap as they were looking at reversing checkerboards or listening to tones. The main point here is that you find a very similar change in entropy. The curve is shorter because the time series length was shorter in this particular data set. But again, the point being is that you have the youngest infants here, the 1 to 2 month old. You have the 60-month-old infants in the magenta, and then not a great control group, but we have young adults as well in here just for metric. So again, entropy increases as you go forward in time. So we found that this information processing capacity increases with maturation, which is kind of what you'd expect, right? You do stuff differently now than when you were 1 month old, I hope. So, and hopefully it's better than it was when you were 1 month old. So, And I did mention that the dominant changes across all time scales. So even though I made a big point about time scale, it looks like, at least in terms of early in life, most of the changes are actually happening across all time scales. What happens in aging? So here I have two different data sets, one EEG, one MEG data set. Again, the tasks are not really that important because the effects are pretty much the same. If we look at EEG data set has three groups, young, middle, and old, whereas the MEG has just young and old subjects. We're going to look at the MSE curves again. In the EEG data set, you find an interesting difference, that the age-related changes are in fact timescale dependent. That at fine scales, you actually have higher entropy in older adults, and it crosses over where at the coarser scales you have lower entropy in the adults. So more information integration at fine scales and less at coarse scales in the older adults. There's also some spatial dependence on that. These are source space projections. You can't read that, but primarily you get differences in precuneus, prefrontal cortex, and some of the motor regions as well, shown in red. That's just basically telling you where in time and space the differences manifest. So unlike maturation, where you have a general increase, in aging you actually have a time-dependent change in information processing capacity, as well as spatial scale dependent. MEG data, again, two groups, exactly the same pattern. You've got a higher entropy in fine scales for the older adults and crosses over to be lower at the coarser scales in the older adults. The spatial pattern is similar, although it's much more broad in the MEG data, suggesting a stronger effect, but nevertheless, you do see the timescale dependency in both data sets. So that's interesting. So it suggests that there's something happening that's time dependent in your brain when you get older. And these are actually fairly healthy older adults. We can relate those changes back to cognitive function. This is a different data set. Here, replicate the timescale dependency again, looking at older adults, again, having higher entropy at fine scales, lower at coarse scales. If we correlate the fine scale entropy to task performance on a Sternberg task and physical activity, we find that if you have higher entropy within the older group, they're doing better. They also tend to be more physically active, for those of us who are into physical activity stuff. If you have more entropy at coarse time scales, which means you don't show that crossover, you actually are doing worse in the task. So by not showing that change, where it goes high at fine scales and coarse at low, you actually have worse performance on the task. There's no effect on the physical activity, or no relation there. So this suggests that there's something about that high fine scale that actually predicts bad cognitive performance. As a matter of fact, we've seen that now in mild cognitive impairment. So here we've got on this side, the left side, if you parse that prior data set into high and low performers, you find that those who are performing low, actually the ones on the very top curve, have higher entropy at the finer scales, which you kind of get from the correlation results. If you look at the far right side, that's actually data from mild cognitive impairment and people who are at risk, and based on their MoCA scores, Montreal Cognitive Assessment Battery for cognitive status. And you find that those who are showing poor health and high MoCAs are the ones that have higher entropy at the finer scales, suggesting again there's something about that lack of switch, if you will, in how the information is processed between fine and coarse scales that means it's bad for cognitive health. As a matter of fact, if we do that in another data set, in Parkinson's dementia, we look at data from people 1 year prior to transitioning to Parkinson's dementia. With three curves there, controls, Parkinson's dementia who did not convert to having, or Parkinson's disease, pardon me, who did not convert to having dementia, Parkinson's disease who did convert to having dementia in the red curve. What you find is those that convert 1 year after being measured, 1 year before actually showed higher entropy at the fine scales, or coarse scale, I should say, again. So something about, again, this lack of change in the balance between fine and coarse timescales that's indicative of poor cognitive performance. So getting back to the notion that space and time is important, we see examples now where we've been able to apply it across different empirical data sets, showing that if I focus on one spatial scale, focus on one time scale, I'd probably miss some stuff. But by taking into consideration that the brain works across space and time, I get much more information about the cognitive status of the brain as well as the potential of that brain to process information. Tough one. So how do we use that? Can we actually do something interesting with this? And this is my last slide. When I was going to put this talk together, most of my talk was going to be focused on the next piece of information. And I decided to not do that because it's difficult for me to understand it. So if I can't explain it, then you're not going to understand it either. But the notion here is to take into consideration that cognition, actually everything the brain does is a process. We heard about motor function, for example, earlier. But everything we do is a process. There's a time point to it and a space point to it and different time scales. So how do we try and pull that together? There are mathematical principles in dynamical systems that allow us to do that, called structured flow on manifolds. My partner in crime in the virtual brain, Victor Jersa, who's the last off in that paper, has developed this method that allows you to take small flows amongst networks across a broader manifold and talk about the transitions between functional configurations. And mapping those transitions across space and time is a great way to characterize the evolution of a process from a system. His application is actually to motor function. Motor function is easy because you can see it. You can see my arm moving, you can see me dancing. Maybe not. You can't see me thinking easily, right? You can measure EEG activity, but you don't know what the process is. So it's really good for motor function. Harder to do for cognition unless we have another measure that tells us the timing of cognition. Eye movements, for example, would be one possibility. So that's the abstract. Don't read it now. The main point is that the basic units in this manifestation are conceptualized as temporally evolving low-dimensional dynamical object or structured flows emerging from a high-dimensional system referred to as structured flows on manifolds. So the structured flows are small networks on a broader capacity of the system to interact. Networks that are forming small configurations that move forward in time across a broader manifold. Kind of like that. Structured flow on manifolds. So the brain works across space and time. There's a concept called timescale separation, which is quite important for allowing these technologies, these techniques, these calculations to be usable. If we model intrinsic dynamics, we can show how the confluence of geometry from anatomy and distance actually and noise allows the brain to explore potential configurations. Modeling that, we can measure that as well. And showing that the space-time structure changes with maturation and can be related to cognitive status. And now I think we get close now to be able to consider mental processes as flows and trying to use the principles I talked about throughout the talk, but also ended off with, as a way to tackle that metric to relate it back to the cognitive processes that we're going to continue to discuss over the next day and a half. Thank you.
So we have 15 minutes again for questions.
Right, that was awesome. I have a gazillion questions, but I'm just going to ask one. So before you talk about all the entropy stuff, you presented a cross-correlation matrix that had, in the diagonal, you were showing areas that are more cross-correlated with each other across different time points,
Yeah.
and some that are less cross-correlated.
Right.
And then you suggested that one of the reasons why it might be possible that they're more cross-correlated across time in certain specific points is because they were exploring some possibilities.
Right.
And I didn't quite get that step, so I wanted to give you an alternative hypothesis as to why there might be a cross-correlation, and I'm going to tell you why you think is wrong, which is, one possibility is that different information might take a little bit long, like different timescales to be transmitted. So if I send you a file that doesn't weigh that much, it just might need a couple of seconds to send that file. But if I send you a file that might require a little bit more time, then I have to spend a little bit more time sending you that file. Why isn't it that maybe you find cross-correlation across different time points because some files are heavier than other files, and so you wouldn't find, so in the parts that are blue, you wouldn't necessarily find them as cross-correlation across times, and what happens in the other ones is that the information that they're carrying is weighs more, as it were.
I don't think you're wrong. I think what you're saying basically is that depending on the information that's being processed, you might get a reconfiguration of the functional networks.
Correct.
Which is exactly what I was saying.
Okay.
I was using a different analogy, using communication theory in packets, but I'm using the idea that there might be different information that needs to be exchanged, and the networks were dissolved and then recombined. You mentioned this morning, for example, when you're looking at the evolution of emotions, there's not an emotional network, but you have bits and pieces being pulled off as the emotion manifests. Just as you have a process going from recognizing an object and being able to act on an object, the networks are going to form and then dissolve as you go through that process. These networks already exist. We know they're there. We've got a lot of data that suggests that there are intrinsic dynamics that result in functional connections. But as you perturb the system, it's going to reconfigure, respond appropriately, usually, and then go back to another state afterwards. So I think we're saying the same thing. It's just different analogies.
Dan?
Yeah, thank you. I was just hoping you could say a little more about different scales and how they might interact. So there's other similar, at least vaguely similar kinds of analyses where people compare correct and incorrect trials on things like memory tasks.
Yeah.
And for instance, prefrontal neurons are high dimensional, so they reflect sort of temporal order and identity of stimuli and memory task conditions. And that dimensionality is decreased in error trials. Similarly, for smaller scale networks, for instance, hippocampal and frontal or parietal networks, decreased connectivity correlates with error trials in memory tasks.
Yes.
So these sound like two different smaller scales. Does this fit into the story at all?
Indeed, and actually you can see that quite well. One of the problems, of course, with being able to measure a lot of the brain is the wealth of data you have to deal with. So one way around that, of course, is just to focus on an area and say, I'm just going to focus on hippocampus and look at the neurons there. But the timestamp there could be quite different than what you see in frontal cortex. And I'm not saying the brain has to reconcile that, but there has to be some reconciliation for the fact that the timescales over which these things operate can be quite different. But that's actually probably an important feature. That's probably how the brain evolved to do what it does right now. It's by having that timescale separation, it allows it to respond optimally to a particular perturbation. Maybe not all the time, but at least in terms of the different timescales that are inherent to an architecture in frontal cortex for hippocampus is going to be important to characterize how it's going to process information as well.
It's really valuable to shift from thinking of mental states and objects to being processes. But at the very end, you suggested we should think of the processes as flows. What's a flow?
A flow is a transition from one configuration to the next configuration. When I was saying dancing is a flow. You typically don't think about dancing as individual steps. If you do, you're going to miss a beat. So the process is actually the movement across time of the configurations of the networks. So that's the flow.
So flow, so configuration of what? In this case, configuration
of the parts of the brain that are interacting at a particular point in time. That's going to be very broad because I'm talking, like what I'm showing you, the second last slide was the entire brain interacting. And that's showing a very broad flow across a very complex manifold, which you can obviously map that down to individual sub-circuits and look at the flows of individual circuits across the manifold.
ask if you have any thoughts about how evidence for the necessity or functional role of entropy comport with evidence for entropy reduction as being functional in the Friston sense. Are these just different concepts, or is there some tension here?
No, not really, because and I actually had some discussions with Carl about that. There has to be a certain amount of entropy in the system always there. It's the question of when it's increasing is actually increasing because of potential error detections, and it's going back to another state. We found, for example, I didn't talk about this, in a study of learning, you find that as people learn more information about, in this particular case, a face. So you learn about it's, this person's married, they had a fight with their spouse, they work at this job, they're going to get, et cetera, et cetera. You get a more semantic elaboration. You find the entropy in response to this face goes up, but then it caps at a certain point. So more information gets acquired, but then it has to go back down. So there's probably a sweet spot. It's not as if it's going to go up continually. It's actually, like most things in biology, it's an inverted U.
What's driving the flow on the manifold? Is it extrinsic, or is it the shape of the manifold itself?
I think when there's nothing else happening, it's probably the shape of the manifold itself that's driving it, and also the configuration of the noise in the system. As soon as you perturb it, you're going to potentially change the shape, at least of the phase flow. It might actually change the manifold as well. Hopefully not. Because the manifold should be fairly deterministic, at least at a very broad time scale.
Yeah, I was just wondering what exactly you meant by noise. Is it just is it a conglomeration of sort of input from the outside world plus intrinsic noise plus variability in individual trials, or is there can you map?
Yeah, there's a really good review article by Wolpert's group in Nature Reviews Neuroscience called Noise in the Brain. And in there, and I don't have the slide unfortunately, there's a diagram of different levels at which noise manifests in the brain. The brain is a biological system, so by virtue of that, it's noisy. Channels open and close inexactly. The timing of action potentials is not necessarily always exact. So there's imprecisions in there. Whether that's adaptive or not is a different issue. So there's noise at that level. There's also noise that reflects the change in synaptic density between neurons locally that happens very fast, as well as distally. It also happens very fast. It's not a huge change, but it's just enough to change the dynamics, so there's additional noise there. That too probably is adaptive. The second level of noise is the exploration, looking at different configurations of the network, which is that reconfiguration in space and time that allows us to explore what's possible. You can parse those in the model at least by explicitly modeling the different levels of noise. Measuring it empirically is a bit more of a challenge. So to incite questions like you just asked, I usually call it noise, and then we have a discussion about different levels of noise. So empirically it might be harder to measure, although there are possibilities, but you can model those different levels quite nicely. You can perturb the system as well, of course, and that will introduce another change in dynamics. It may not be noise per se, but it will change at least the noise profile, at least temporarily, as the transients traverse the system.
I mean, that wasn't clear very early on in your talk when we had the manifold without noise and with noise.
Yeah.
Was that
So with
noise?
Noise is that noise is actually intrinsic to the cell population I'm modeling. So in the situation where I had no noise and noise, no noise was basically an oscillator with no noise term. The noise one actually had an oscillator and then a Gaussian noise on top of that. So that one is easy. It's Gaussian noise, okay. But the rest of it, like later on, as you get more complicated systems, noise is going to be some conglomeration of all these things probably.
Right, so can I just ask a follow-up? I mean, a lot of times in what at least the neuroscience when we talk about noise, it's just unexplained variance. It's not necessarily anything it's not random necessarily. I mean, it could just be inputs from other parts of the system that are not accounted for in whatever model.
Yeah.
Right?
So this is true. The noise is a bad thing in linear systems. Noise in a nonlinear system is actually a good thing to a certain degree. And again, back to the point earlier about entropy, is that in a nonlinear system, having a certain amount of noise is actually good for it to be able to respond. it does lack a bit of precision in terms of its trial-to-trial response. If you have a linear system and you ping it, it's going to do exactly the same thing until you put too much noise in there. A nonlinear system will have a bit of slop in there, but that's actually good for it to be adaptive. You made a comment in your talk about motor systems, for example. And there's a very nice review I was trying to find when you were talking, I couldn't find it, talking about the importance of actually having noise at a low level to allow a very high precision in limb movements, for example. If you watch a golfer, for example, swing, pro golfer swing, you'll find that there are micro movements as they swing the ball towards the ball. You don't see that in amateurs. Those micro movements happen much, much faster than you could possibly think of happening in a particular brain area. That comes because there's noise in the cerebellum that allows them to make the adjustments very, very fast. But maybe even seen that. But that's a micro scale. But that noise is quite important. And it's not noise is the bad thing there. Noise is actually good because then they get, to wear the green jacket, as it were, at the end of the tour.
Question. You do focus on the term structured flows on manifolds, and so I have a question, and maybe others do with it. If I could put the spotlight on the term flows and ask for clarification as follows. It seems like you have a dynamical system which is in a certain state, and then it's making a transition to another state. And that sounds to me like a kind of a discrete process. Is it the transition from one point in phase space to another point that is what you mean by the flow? Or,
yeah.
Or am I missing?
If I may.
Because when you showed that diagram, exactly that one. I mean,
I'm going to show you, this is actually what I'm talking about.
Okay.
So you probably can't see this, unfortunately. So what I'm showing here across time, and the bottom is the attractor space for the system.
Yeah.
And you see it actually going from a cyclic to a fixed point attractor in the middle.
Right.
So the transition actually happens very fast in the crossover point in the middle. It changes the flow within that part of the manifold, and then we'll at some point transition back spontaneously. So it has a slow variable and a fast variable. Slow variable is the broader phase flow. Transitions happen very fast, the bifurcation, and then it goes to another configuration, and then back to another configuration afterwards.
Okay.
So those are the flows.
Transitioning, that's the flow. It's at the stable point, then that's characterized by that
flow. And there's
different.
there's actually flow the whole thing, I would call it. But that it's the transition as well as it's just a question of how fast do those transitions happen, how fast do the flows happen. There's a slow part of the flow in the initial part of exploring the phase space. When it gets close to the bifurcation, it's fast, and then it transitions to a different configuration.
Okay, well, I think time's almost up, and maybe I think we're going straight into the next speaker,
Yeah.
but maybe we can take like a 2-minute bathroom break. That definitely needs to
Bio
go.
break.